Hello, I'm a PhD student in the Australasian Wildlife Genomics group, and today I'll be talking to you about a study where I compared the reliability of genotype calls at both MHC and TLR genes using two different methods, whole genome sequencing and target enrichment. In this study, we investigated 10 koalas from a population in the Moreton Bay region, which is in southeast Queensland, using two different methods. The first method was a target enrichment probe, which targeted 1,210 immune-related genes. And this is part of a project investigating genomic associations with resolving a chlamydia infection in 43 different koalas. We also used a whole genome sequencing approach of at least 30x coverage. This was part of a larger genome survey of 400 koalas in Australia. We chose two commonly investigated immune gene families to assess the genotype reliability on, toll-like receptors and the major histocompatibility complex. Our target enrichment sequencing achieved an average depth of 250 times across the target region, and we therefore used these genotype calls as a truth data set to compare our whole genome data set to. We also downsampled our whole genomes to a range of sequencing depths from 0.5x to 30x coverage. The MHC is a vital part of the adaptive immune system and responsible for binding peptides and presenting them to T cells. Class 1 genes undergo rapid expansion and deletion between species, and they're phylogenetically distinct. We identified 19 class 1 genes in the koala genome. Class 2 genes, on the other hand, have a slower evolutionary rate than class 1 genes, and they share orthology across clades. And marsupials have a DA, DB, DC, and DM molecule, which is encoded by an alpha and a beta gene. The number of copies of each of these genes varies between species, and in the koala, we found a single copy of the DA alpha as well as both DC and DM genes. We found 3 copies of both DB genes and 5 copies of the DA beta gene. The peptide binding region of the MHC shows incredible diversity within species and populations, and it's the diversity within this region that allows the MHC to bind to a broad range of pathogens. High levels of diversity within this region of the MHC increases the fitness and genomic health of a population. However, this provides technical challenges to researchers studying the MHC, and class 1 in the koala have nucleotide similarity sequences of up to 95%. The high level of diversity within the MHC region is shown with our 10 samples having 301 different variants. Toll-like receptors form part of the innate immune system, and the genes which encode membrane-spanning molecules which bind to conserved molecules expressed from the pathogen surface, known as PAMP. Once the TLR gene binds to a PAMP, a cascade of genes involved with inflammation and immunity are activated. TLR genes are ancient and conserved genes which are shared across vertebrate lineages, and there are 10 TLR genes in marsupials. 9 of these show direct orthologs with the Eutherians, and one gene, the TLR16, is an ancestral gene of the TLR1 and TLR6 genes seen in Eutherians. For this study, we used TLR genes as a proxy for conserved regions of the genome, which is shown by the fact that only 28 variants were identified within our TLR genes in the 10 koalas. PCR followed by Sanger sequencing is a common method to investigate the genetic diversity in a single or very few genes. This method is popular as the sequence is read directly and has extremely high base call accuracy. However, once you begin investigating multiple genes, this approach is no longer cost-effective. Next-generation sequencing has dramatically decreased the cost of obtaining sequence data. However, these technologies have lower base call accuracy and are limited by a read length of up to 250 bases. This means that often the reads will have to be assembled in order to investigate the genetic variants. The assembly of these reads occurs through alignment to a reference genome, and for most of the genome, these reads will map to a single location. However, there are portions of the genome that are highly repeated or duplicated, which makes alignment difficult. This is commonly seen in MHC genes, and most studies that utilize PCR followed by next-generation sequencing investigate only 1 or 2 exons of the gene. This is most commonly the peptide binding region, as this is where the majority of the variation is found. The peptide binding region is encoded by exons 2 and 3 in class 1 genes and exon 2 in the class 2 genes. As the utility of genomics increases in conservation, more broad-scale genome-wide techniques are being used to investigate not only mutual diversity but functional diversity. And it's vital that we understand the level of sequencing which is required to accurately genotype important functional regions of the genome, such as the MHC. Looking first at the TLR genes, at coverage below 2x, we can see that the majority of non-concordant calls is due to missing data, represented by the blue line. However, between 2 and 5x coverage, we can see a decrease in the missing calls from 32% down to 5%. And at 10x coverage, there is no missing genotype calls in the whole genome dataset. Also, at 10x coverage, we are able to see that less than 10% of variant calls are incorrect, which is shown by the yellow line. And out of the 27 incorrect calls, 19 of these occurred in a single individual, suggesting an issue with that sample. We're able to see a plateau in the percentage of correct genotype calls, shown by the orange line, between 10 and 15x coverage. And at 15x coverage, there are only 20 incorrect variant calls, all of which occur in the same individual. We can also see that by 10x coverage, all of the genotypes identified with the target enrichment are also seen in the whole genome sequencing. We therefore suggest that between 10 and 15x coverage is sufficient to reliably genotype most regions of the genome. The same as TLR genes, below 2x coverage, the majority of non-concordant calls in MHC genes is due to missing genotype, which is shown by the blue line. However, by the time you have 30x coverage, this reduces to less than 1% of the genotype calls. The percentage of correct genotype calls, again shown by the orange line, plateaus at around 15x coverage at close to 85%. Interestingly, also at 15x coverage, we see a number of genotypes called in the whole genome dataset, but not present within the target enrichment, which is shown by the black line, which could be due to mapping issues in the whole genome sequencing. Also in MHC genes, we see 84 variant locations present within our whole genome dataset that are not identified by the target enrichment set, which is shown by the blue bar in this plot. We found that 15 of these variants were initially present in our target enrichment set, however, were filtered out for various reasons. 75 out of 84 of these variants were located within class 1 MHC genes, and 22 of these sites were located within the UG gene, which had no reads in the target enrichment map to it, despite this area being targeted by our probe. There are an additional 46 sites which were in the genes UH and UB, which indicates that the sequence similarity of class 1 genes to one another makes these genes far more difficult than the class 2 genes to genotype. And in the koala specifically, the UB, UG, and UH genes can be difficult to align sequence reads to. This is supported by high sequence similarity between these three genes, with nucleotide similarities of up to 90%. The study shows that 10x coverage should give enough depth to reliably genotype most regions of the genome, and that duplicated and repetitive regions require higher sequencing depth at 20x coverage or above. We also found that MHC class 1 genes are more difficult to investigate than the class 2 genes, and potentially long-read sequencing may be a better use of resources than the whole genome sequencing if the aim of the study is to directly investigate MHC genes only. I'd just like to thank all the people involved with this project. I'm happy to take any questions.